Let's see. Boggs is here. Bonner is online. Clay is here. Green? No. Harrison's here. Vincent's here. Esterson's here. King? Yeah, King is here. Morrison is here. So far, everybody's counting. Wilder is online. So when you guys, if you ever decide to attend virtually, streaming it, make sure you're signed into your WVSU email account. Somebody was just trying to get in, and it won't let people in unless you're signed into your WVSU email account. Or if I physically let them in, but can't always do that. All right, Ward is here. And then Green, right? Yeah. And we've got about 5 minutes, so if you guys have any questions, let me know. I'll just do the usual reminders. Make sure you've done your pre-lab. If there's any assignments you've missed, go ahead and turn them in. Better late than never. I didn't do any work this weekend. I didn't read any emails, didn't grade anything, so I have a lot of catching up to do. And I'm feeling under the weather, so it might be tomorrow before I'm caught up with grading everything from last week. But I will let you know. I'll post an announcement once I am. And then at that point, once I'm done grading everything from last week, if you have any questions about your grades, let me know. What else? A reminder, you can meet me online to go over your old assignments to get feedback. Any assignment that you do that with, you get a 5% boost to your grade. Sometimes that's not necessarily true. So if, for example, you score perfectly on a quiz, we don't have much to talk about because you got everything correct. If it's a lab and you scored perfectly, I might be able to give you feedback, because with labs, there's a difference between you got all the points or you did perfectly, right? So very often people get full credit, but it wasn't perfect. So I can definitely give some people some feedback despite perfect grade. This is it for reminders. It's pretty much on autopilot now, right? You guys know how the course works. We're just going through it. James is online. All right. So for those of you online, as a reminder, you have to send in the keywords, the attendance words. Just online doesn't mean you're counted as present. Morning. Dawson? Sorry. Key, right?
Dawson.
No, yes. Need my coffee to kick in. Morning. So Parks is here. Linda Q is here. Excuse me. So you guys won, right?
Yes, sir.
Yeah, on the road too. Glad one of my teams won. Actually, my other team didn't even play this weekend, so for once, Florida State didn't have a loss over the weekend anyway. All right, you've got Wilcox this year. With less than a minute left, I'll go through the tenants from top to bottom. Aki, not here. Lount, not here. Boggs is here. Bonner's online. Clay is here. Clayton, not here. Dawson is here. Ellis, not here. Tala, not here. Green is here. Harrison's here. Henson's here. James is online. Kesterson is here. King is here. Lewis is here. Linda Q is here. Morrison is here. First name Grace, not last name James. Not here. Rice, not here. Ward is here. Wilcox is here. Wilder is here. Parks is here. Did I miss anybody? All right, let's jump into it. And again, like I said, I'm really feeling under the weather, so I apologize in advance for being a little bit slow. But to remind you what we were talking about, again, this portion of the chapter is all about biological molecules. So the big four categories, I guess I should say big four, because usually when I say big four, I'm talking about oxygen, hydrogen, nitrogen, and carbon. But the four categories of biological molecules that we were talking about are carbohydrates, lipids, proteins, and nucleic acids, right? So we finished talking about carbohydrates, then we started getting into lipids. The last thing I told you, well, other than this slide, is that lipids are different than the other ones, right? So with the other ones, it's all about, what is the monomer, what is the polymer? In this case, they're not necessarily macromolecules, so they don't necessarily have repeating monomers, so there are no polymers. But again, the lipids are made up of different pieces, right? So for example, this is a triglyceride, and it's made up of pieces, right? You have the glycerol, and you have 3 fatty acids. Now later, I'm going to come back and talk about the structure, because again, and this seems to be a theme with this part of the chapter, there's a very strong correlation between the structure and the function, right? So this shape will actually mean something, and we'll talk about that later. Before we talk about that, though, can someone guess, if you had to, and I think Kesterson can get at least one of these, because he asked a question about this last week. Oh, never mind, it's right there. I was going to ask you to see if anybody could guess, but there it is. The essential functions of triglycerides are energy storage, cushioning, and insulation, right? So Kesterson was asking again about burning calories. When we were talking about carbohydrates, I said that's the main source of energy for animals. But then he asked, well, what about burning fat? And that's what this is, right? So we do use fat to store energy. Obviously cushioning, which is why I chose this picture, right? You've got fat on the bottom of your feet. You also have fat other places for cushioning, but I didn't feel like putting a picture up there would be appropriate for that. And insulation, right? So maybe not so much humans because we have clothes, but you think about walruses, right? Big and blubbery. And that's one of the reasons why. So any questions about the essential functions of a triglyceride?
Do fish have fat?
Yes, that's a great question. And some more than others. But I'm not an ichthyologist, so I'm not really familiar with it. But yeah, they do. For example, salmon, I think, are fattier than others. I think. Great question. That's also a good independent work topic. And now I'm curious, is there a correlation between where the fish lives and how fatty it is? Like cold water fish, I wonder if they're fattier. But to your point, and I think I'll actually bring this up a little bit later, they definitely are. There are some that are fattier than others, and they're good fats. Like they're called omega-3 fats. They're really good for you, and that's usually one of the only places you can get them. So yeah, not only are they fat, do they have fat, but the fat that they have is good for our diet. Which brings me to this point. There is 2 different types of triglycerides, and you should know the difference between them for the exam. There's saturated and there's unsaturated. Long story short, the unsaturated, well, I don't know if I want to say that yet. Yeah, I won't say that yet. I'll come to that. So unsaturated. What makes it unsaturated? Or first of all, no, let me go back. saturated. What makes it saturated? This is something you're going to need to know for the exam. If we weren't talking about fatty acids, right? Let's say I was just out for a run and I came back and I said, oh man, I'm saturated. My shirt is saturated. What do you think I mean? Sweat, right? So much sweat that my shirt can't absorb anymore, right? That's what we mean by saturated, right? It's as much as it can hold. Same here. So these triglycerides, you remember there's 3 of them that are attached to the glycerol molecule. If they're saturated with hydrogens, that's what we mean by saturated, right? They have all the hydrogens that they can carry if it's saturated. If it's unsaturated, they don't have all the hydrogens they can carry, right there. And that's just one example. So you definitely need to know that for the exam. What are saturated fats saturated with? Hydrogens. And then beyond that, why are unsaturated fats unsaturated? Why don't they have as many hydrogens as they can? And this is something else you need to know. It's because they have double bonds. So those double bonds change the shape. You can see that one up top is straight, right? And you see the one below has like a little kink in it, right? And that is actually important at the molecular level. And again, I know none of you are chemists, but imagine, like that is the shape of the molecule, right? So imagine trying to stack a bunch of saturated fats on top of each other versus trying to stack a bunch of unsaturated fats on top of each other. And also keep in mind, this is just one unsaturated fat. So that little kink could be here, could be here, could be here, right? They're not always right there. It could be any place on the saturated, saturated fat, which is unsaturated. And these things can be longer, they can be shorter, so the size changes as well. So again, what do you think would be easier to stack? A bunch of saturated fats or a bunch of unsaturated fats? Saturated, yes, because look, it's flat, you can put them on top of each other. And that is why molecularly, that saturated fats are worse for you, because that's exactly what they do. They just kind of stack up in your bloodstream, right? So that's how you get clogged arteries, because they're stacking up on each other, because molecularly they fit that way. Anyway, really quickly just address these bullet points. Again, unsaturated fats, the fatty acid doesn't always have the maximum number of hydrogens. We often call them oils. I'm going to put an X to that because I'm not going to test you on that. It's true, but not going to be a test question. They are liquid at room temperature, and again, molecularly, the reason that they are liquid at room temperature, again, we're talking about unsaturated. I should probably flip those on this PowerPoint. Anyway, the reason they're liquid at room temperature is because they don't stack nicely, right? So instead of sitting still, they're liquid. Unsaturated fats are generally healthy. And then we have something called polyunsaturated fats, which is where they have several double bonds. So again, said this one example, there's one double bond right there. Sometimes there's more than one double bond. So that's it for unsaturated. For saturated, to be a saturated fat, all 3 of the fatty acids have to be saturated with the hydrogen. They're generally solid at room temperature, and they're generally unhealthy. So any questions about these distinctions between saturated and unsaturated fats?
Does this apply to like when you go buy vegetable oil, it's say saturated or unsaturated?
It would, yeah, I imagine. And you know what? I've never paid attention on the vegetable oils, whether it says saturated or unsaturated. I've never paid attention to that. But, you've got things like Crisco. It's vinegar. I was hoping that was a little. Anyway, you've got things like Crisco, right? So that's solid at room temperature. Or how many of you ever cooked bacon and it's all liquidy and fatty, right? And then what happens when it cools?
Solid.
Yeah, it becomes solid, right? At room temperature, it becomes solid, right? So, and that's why. And again, that's also why it's bad for you. But, that's stuff you can look in. That's a good question. You can look up that for individual work.
So you're saying bacon fat is unhealthy for you?
Yeah, sadly. Sadly it is. Anyway, any questions about this slide? Yes, ma'am.
To confirm the differences that saturated is over with hydrogen.
Mm-hmm. Yeah, when we say saturated, we mean it's saturated with hydrogens, right? It's all the hydrogens it can take. Any other questions? All right, this is not from your textbook. This is from the old textbook, but I wanted to include it anyway. Usually it's animal fats, right? So when we're talking about saturated, it's usually animal. But not fish, and we'll get to that later since you already asked. So yeah, most animal fats are relatively high in saturated fat. Again, they can easily stack at room temperature. And this is why we get clogged arteries, right? Because they just fit together nicely. Any questions about that? All right, so if most animal fats are saturated, most plant fats and fish fats are actually high in unsaturated fat. It's usually liquid at room temperature, and tropical plants are the exception, right? So avocados, coconuts, those are the exceptions. I'm probably not going to test you on that. But if I did, again, you should associate animals generally with saturated fat, and you should associate, other than fish, and you should associate fish and plants with unsaturated fat. Actually, that brings up another good point. This is from your old textbook or the new one? Anyway, obviously we're talking about diet there. So when we say animals are high in saturated fat, I mean the diet, right? So red meat, things like that. But now I'm curious, I honestly don't know the answer to this. I wonder if all animals have that. For example, sponges are animals, corals are animals, and can't picture they would have much fat, whether it be saturated or unsaturated, and I honestly don't know the answer to that question. So if you want to look it up for independent work, go ahead. And I also will say that, no, we'll come to that later. There's other stuff, but we'll get there. Anyway, any questions about that side? Here's another thing that's not from your textbook, but I included it anyway. So you can see these different corn oil versus palm oil versus butter. And Kesterson was asking about when you buy oil from the grocery store. So there is a good example. Corn oil is what, half polyunsaturated, which is good stuff. 37% monounsaturated, which is good, but not as good as polyunsaturated. Then of course, 13% saturated. I'll put a big old X through this because I'm not going to test you on this. And look, that's just 3 different types of oil. So once again, if you're interested in that stuff, you can look it up for independent work. What about canola oil? What about olive oil? Right? All these other different types of oils. Also, they have different smoking points. So if you're into cooking, that's something else you can look into. At what point do they smoke? Which one has more calories? All those things you can look into. All right. Then there's this process where we can convert unsaturated fats to saturated. How do we do that? Well, obviously, if saturated means it has all the hydrogens it can, then the process is giving it hydrogens, and we call that hydrogenation. I'm going to put a question mark next to this. I might ask, I might not. That's obviously pretty relevant to your everyday life because you might be eating something that has been converted to a saturated fat. But as far as the exam is concerned, as far as the semester is concerned, that's not necessarily very important information because we'll probably never talk about this again, but there it is. The process is called hydrogenation. The reason they do this, among other reasons, it doesn't spoil as quickly, and I don't know why that is either, so that's something else you can look up from independent work. What is it about being hydrogenated that makes it spoil less quickly? Why does it preserve it? It's good for frying. Can be solid at room temperature, like margarine, right? That's a good example. But it can create something called trans fats, and trans fats are very bad for your health. They're found in margarine, some peanut butter, and it increases the bad cholesterol, which is LDL. So if anything, for the exam, again, I probably won't ask about hydrogenation, but maybe. So maybe know that hydrogenation is the process of converting unsaturated to saturated, and maybe know that trans fats are very bad for your health. I'm not going to ask you which is the good cholesterol, which is the bad cholesterol. Just again, if anything, know what hydrogenation is and know that trans fats are very bad for you. And again, another good independent work topic. Why are trans fats so bad for you? So let's talk about a different type of fat. And we started talking about this because Kesterson was asking about the fish. So there's something called omega-3 fatty acids. These are essential fatty acids. And in this class, when we're talking about biological molecules, when I use the word essential, what I mean is we can't make it, right? So when they talk about like essential vitamins, it's vitamins that we can't make. Or in this case, we cannot make that kind of fatty acid. So because of that, we have to get it through our diet, which is why it's good to eat fatty fish like salmon, as an example. And nuts, right? Walnuts are high in omega-3 fatty acids. And again, more good independent work topics if you're into it. So when I say it's found in oily fish, obviously I already told you salmon. What are some other examples of some oily fish? Which fish have the most omega-3s and which nuts have the most omega-3s. Yes, sir.
What about this pill they sell that's called fish oil?
I take that, yeah. And supposedly it's good.
Yeah.
If it's true. I've also heard too that a lot of supplements are just BS because they're not regulated. So I could be just taking who knows what. But it has helped my health, so I assume that the particular brand I'm using is actually going to be fish oil. But yeah, that's exactly what it is.
You need to take zinc too.
Yeah, I take that sometimes too when I'm feeling sick.
It'll cut down the longevity of a cold and stuff.
Yep. So, any questions about omega-3 fatty acids? All right. So again, like I said earlier, and I was about to get to this and I stopped myself, and now I'm going to go ahead and do it. I was asking, do all animals have fat, right? So again, coral is a great example. Sponges, a great example, I think, because again, those are technically animals, and I just can't picture them being very fatty. And then I remembered, and this is where I'm getting at here, everything that has a cell technically has fat. Because, and this is another type of fat, right? So now we're done talking about triglycerides. Now I'm moving on to the next subcategory, and they're called phospholipids. You definitely need to know about phospholipids. That's what makes up our cell membranes. And again, if you remember from chapter 1, those properties of life, one of the things that's a property of life is cellular, right? So everything that's alive is cellular. Therefore, it has a cell membrane, and therefore it has this type of fat. And it's similar to a triglyceride, because remember, a triglyceride is that glycerol molecule with those 3 fatty acids attached. And in this case, it's 2 fatty acids in a glycerol, and they're attached to a phosphate. I might ask you that. Put a question mark next to that. I'll put a check mark next to this one because you definitely need to know phospholipids make up our cell membrane. Of course, there's other things in our cell membrane too, but we'll talk about that when we get to that chapter. But yeah, the majority of it is phospholipids. And they're interesting because they are both hydrophobic and hydrophilic. So the fatty acid parts are hydrophobic, and the other part, the glycerol and the phosphate too. The phosphate is the hydrophilic part. So I'll put a question mark next to that too. I might ask you that. I won't ask you to remember which is which though. I won't say, in a phospholipid, which part is hydrophobic and which part is hydrophilic. I won't ask you that. If anything, you should know that phospholipids are both hydrophobic and hydrophilic. And I'll go ahead and tell you now, that's the only thing in this section of the chapter that's going to be both hydrophobic and hydrophilic. So that makes it unique.
The meeting in the middle and then the hydrophilic ones are kind of in the same. I don't think I talked about that. Is that what he's talking about?
Sort of, yeah. So the hydrophobic parts, like if you were looking, you're going to really get familiar with this next chapter of the cell membrane. It's called a phospholipid bilayer. There's actually 2 layers of it, and the hydrophobic parts stick into each other like that. And the outside is hydrophilic. So hydrophobic on the inside, hydrophilic on the outside, and that kind of makes sense, right? Like think about a cell. Sometimes water can come in and out. So I'll just leave it at that for now, because we will get to it next chapter. So spoiler alert. Any other questions? All right, this is a picture not from your textbook, but once again I liked it, so I kept it in. So again, generally speaking, for the exam, when it comes to saturated fats, you should think unhealthy. Generally speaking, you should think of animal products. And for unsaturated fats, you should generally think of healthy plants and fish, right? And of course, there are exceptions. And the exception is there, right? Margarine is not healthy. Obviously, trans fats are not healthy. But yeah. Anyway, any questions about that? All right. So again, we're still talking about lipids, right? This is a big category. We're done talking about carbs. We talked about that last week. This week we're talking about lipids. And we're done talking about the phospholipids and the triglycerides. Now we're moving on to another category of lipids, and that's called steroids and laxatives. Oh, man. So the first word for attendance, took me a while to get there. Let's say is, what's his name? Who's that guy that got in trouble for steroids in baseball? Well, there's a few. Let's just say baseball. Let's keep it simple. Baseball is the first word for attendance. Damn, what was that guy's name? That headed guy, you guys probably weren't even alive.
Alex Rodriguez,
He probably did too, but that's not who I was thinking of.
Ray Bonds,
No, but again, at the same time, same time frame.
Mark McGuire.
Yes, yeah, Mark McGuire, he was redheaded, right? Yeah. All those records are broken when I was in boot camp, and not all of them, but a lot of them. Actually, yesterday was the day that I went to boot camp, way back in 1998. Anyways, steroids, let's talk about them. Steroids are hydrophobic because, again, they're lipids, and other than those phospholipids we talked about earlier, you should definitely associate lipids as being hydrophobic. They're different than fats, right? So technically they are lipids, but they're different than the fats that we talked about. They're different than those triglycerides, right? They have a different structure. Therefore, again, they have a different function, right? It's all about structure and function. I probably won't ask you this, but not definitely. Their structure is a carbon skeleton that has 4 fused rings. And that makes sense on the next slide when I show you a picture of it. But they have rings, right? So far, that's not the only thing. It's one of the only things I've talked about that it has a ring as a structure. And again, it'll make more sense when I show you a picture. They vary in functional groups. Yeah, I'll just go ahead and show you. That's what it looks like. So this is the basic steroid skeleton, right? All steroids have this basic shape. But it's the functional groups that come off of them that make them different from each other. How many of you guys know of any kind of steroid, like the names of them? Yeah, prednisone's one, right? You get, like if you get bad poison ivy, for example, you might get prescribed prednisone. What's another one?
I think eye drops has got steroids.
Oh, wow. Okay. I forget the kind my daughters take. She has asthma. And it's just, like I said, I'm just tired. That's a antibacterial, actually. But, what is that stuff? Anyway, she takes it for her asthma, and it actually really jacks her up. She gets all angry. She's a mean little kid when she's on it. But anyway, yeah, so steroids, that's the basic function. Another good one, and I'll give you this example in a few slides too, but think about like, which steroid, generally speaking, makes a man a man? Generally speaking, a male a male, I'll say that instead. Testosterone, right? Yeah, and which one generally makes a female, generally? It's not that cut and dry. I am making it a little bit easier than it actually is, but what's the female one? Estrogen, yeah. And again, whether it's testosterone or estrogen, they have this shape, or prednisone, and the other ones we talked about, right? They have this shape. Then there's something called cholesterol. Cholesterol is a key component of cell membranes, and it is the base steroid. So that picture I just showed you, you could basically consider that cholesterol. So what happens is your body puts on these functional groups, right? It changes it, right? So we can start with one, cholesterol, like you can see up here, that's what cholesterol looks like. And when there was some slight changes, you can either have testosterone or estrogen, amongst others. Those are just 2 examples. Now think about that. Look at the structure. And again, none of you are chemists. I'm not a chemist either. But you can see how similar they are, right? Those 2 things look very similar, but they are slightly different, which is, to me, very fascinating because obviously there's a lot of differences between males and females, well, depending on the creature you're talking about, but usually. And look, there's not much difference. Again, here we have an oxygen that's double bonded. And here, instead of being double bonded, that oxygen is attached to a hydrogen. What else? Methyl group. Oh yeah, and then once again, a double bonded oxygen there instead of a single oxygen and a hydrogen. Not that I'm going to ask you that, but just look at what a small difference that is. But then think about what a big difference it makes in things like lions and humans. Anyway, any questions about steroids or cholesterol? All right. So your old textbook mentioned the kind of steroids that I used to think of when I think of the word steroids outside of this class. When I think of steroids, I think of the type that people abuse. Those are called anabolic steroids. This is not, again, not from your textbook. This is from the old textbook. So I'm not going to ask you anything about this, but might as well talk about it very quickly. So these anabolic steroids are basically variants of testosterone. They mimic some of its effects. We use them to treat diseases such as cancer and AIDS, right? Anything where your body's wasting away, not anything, but if there's a disease that's causing your body to basically waste away, then a lot of times steroids are prescribed for that reason. As we know, when I brought this up earlier, there he is. That's the guy I was trying to remember. Sometimes steroids have been used or abused to build up muscles quickly. And again, none of this is in your textbook. I'm not going to ask you any questions about this. Any questions about synthetic anabolic steroids? All right. That brings us to yet another category of lipids, and it's called wax. This is a hydrocarbon chain that has an alcohol group attached to it and a fatty acid. So once again, right, like I said when we first started talking about lipids, it's not necessarily macromolecules with repeating monomers, but they do still, they're made up of parts, right? So once again, we have a fatty acid involved, but in this case, we also have an alcohol group. I'm definitely not going to ask you, I'm not going to require that you know that the OH is an alcohol group. I might just ask you, but you might need to know that wax does have an alcohol group and a fatty acid. And to remind you, multiple choice exam, freshman level, non-biology majors. So there will be no question where I say, explain what a wax is, right? But I might explain what a wax is and then ask you what type of molecule is this, right? So I might give you this description, including this, right? Some examples of beeswax and lanolin. Obviously, I guess I wouldn't give you that example in the test because if I ask you, if I say, all right, this molecule, an example of this molecule is beeswax. It's a hydrocarbon that has an alcohol group attached to it and a fatty acid. What is it? If you miss that, that would be pretty sad, right? Because it's right there in the name, wax. So yeah, if I were to ask you questions about it on the exam, I probably wouldn't use beeswax as the example. That's a little bit too easy. Plants also use wax to protect the leaves, mostly from, well, let's leave it at that. You can look it up for independent work. What do they need protection from? Right? What are the plants protecting themselves from? Any questions about wax? All right. Any questions about lipids in general? Because now we're done talking about that whole category of biological molecules. And that means we're going to move on to the next one, which honestly is my favorite of the four. Proteins. Proteins are very diverse. And remember, like I told you, a lot of this exam is going to be differentiating between the four different types of biological molecules. And I told you one of the ways that we're going to do that is talk about the monomers and the polymers. So here's something you definitely need to know is the second bullet point right here. Proteins are polymers of amino acids. Another way of saying that is the monomer of a protein is amino acids. So you definitely need to know that amino acids are the monomers of proteins. So far now you've been, not familiarized, but I told you about two different types of monomers and polymers, right? First we talked about carbohydrates. The monomer was a monosaccharide, the polymer was a polysaccharide. And here, now I'm telling you the proteins, the monomers are called amino acids. And I'll tell you what the polymer is later. So cells, when you take all the water out of it, 50% of the weight of it, it usually comes from proteins, which should tell you something, right? Obviously, it's very important because most of your cells, other than the water, is made up from this. And they are instrumental in almost everything cells do. And this is why it's my favorite category. Because everything we learn up until the end of the semester is going to be about cells, right? We're going to talk about how cells do this thing and that thing. And everything they do is due to proteins. Proteins pretty much run the show. So they're very important. They are the most sophisticated molecule in the body. None of that's going to be on that exam. So let me think. For the exam, again, definitely know that bullet point. Yeah, that's it. The rest of this is more like those other bullet points are like, hey, this is why we're talking about them. This is why they're so important. But probably not going to be test questions. Here's a little chart that's not from your textbook. It's from the old textbook, but I included it. There's different categories of proteins. I'm not necessarily going to test you on them, but I like this. This is nice, sums it up in one picture, right? So you have stuff like structural proteins. So her hair is a structural protein. Fingernails is a structural protein. Those horns are a structural protein. Then you have storage proteins, right? Things that are like in seeds and eggs. So it's storing amino acids. So things can grow, like baby plants or baby animals. Then there's contractile proteins. So I think probably when most people think about proteins, that's probably what they think about. Like if we weren't in biology class and I said, I'm gonna go get some protein, what do you think I mean by that? Or what would you, or someone said that to you, what do you think it would be? Like, I'm gonna go get some protein. Yeah, maybe that too, yeah. I didn't even consider that. Yeah, that would make more sense. Not what I was getting at, but yeah, you're probably more right. I was thinking like, I'm gonna get some protein, like I'm gonna go, eat a steak or something, go eat some meat. And that's what contractile proteins are. And then there's transport proteins. Those will be really important for us later in the semester. We're gonna talk a lot about transport proteins and we're gonna talk a lot about enzymes. So those two on the right, as far as this semester is concerned, those are the most important ones. Again, as far as the semester is concerned, because we're gonna talk about those, but as far as life is concerned, they're all equally important. Anyway, any questions about that little table? Yes,
Can you please remind me what an amino acid is?
that is the good question. That is the monomer. So that's what proteins are made up of. I feel like you might be psychic because, there we go, next slide. Proteins are made up of 20 different amino acids. So that's something you definitely need to know. I'm not really big into numbers, but that's a number you need to know. Every single protein in your body is made up of 20 amino acids. And I don't mean like you put 20 of them together and you have a protein. What I mean is like there's 20 building blocks to choose from, right? So again, I picture like a Lego set and you have 20 different types of Legos. You might put together a Lego set that has thousands of pieces, but you only had 20 types of pieces to choose from. Does that make sense? That's what amino acids are, right? There's 20 different types of amino acids. Actually, you could look that up for independent work too. There are actually other amino acids, but when we're talking about proteins and how they work, those are the 20 amino acids that we need. Every amino acid consists of a central carbon atom bonded to 4 covalent partners. You should definitely recognize this picture as a monomer, an amino acid. If I were to show you that picture on the exam, you should be like, oh yeah, that is an amino acid. And here's where it gets interesting, or complicated, depending on how you look at it. Every single amino acid has this shape, and they're attached to these things. And this is the only variable part. It's called the side chain. So again, like I said, there's 20 different types of amino acids. And the only thing that makes one different than the other is what they have here, the side chain. Because they all have a central carbon. They're all attached to an amino group. They're all attached to a hydrogen. They're all attached to a carboxyl group. But it's the side chain that makes it different. I will also, though, go ahead and put an X to this for you. I'm not going to ask you to know the difference between an amino group and a carboxyl group. You don't need to know that. But you do need to know, again, if I were to say, if the test question was asking you about what kind of molecule it is, I'd be like, well, it's the central carbon that's attached to an amino group and a hydrogen and a carboxyl group and a side chain. You should recognize that as an amino acid. Again, I'll not give you a question where it says, tell me what an amino acid is attached to, right? I won't do that. Any questions? I'll soon talk a little bit more about this, but are there any questions so far? All right, so again, everything I just said, now I'm going to put it in writing. There are 3 groups that are in common to all amino acids. So you've got the carboxyl group, the amino group, and the hydrogen atom, right? So that carbon, that central carbon, for all amino acids, is attached to the carboxyl group, it's attached to the amino group, and it's attached to a hydrogen atom, right? No matter what the amino acid is, they all have that in common. So once again, this is everything I've already told you, now it's just in writing. The variable component of the amino acid is that side chain, or R group. And once again, when you're studying this, if this is overwhelming to you, remember, it's a multiple choice exam, so I'm not going to ask you to spit all this information out. It'd be the opposite. In the exam, I'll probably give you all this information and say, what am I describing here? You're like, oh, I recognize that. That's an amino acid. I guess acid would be the next word for attendance. Any questions about this slide before I show you another picture of an amino acid? Oh man, this is a really great joke. But it's not, it's really inappropriate. So I won't say it now because we're going to record it, but after I stop recording, and if you're interested in hearing a really dirty joke about amino acids, let me know. It's very rare that you get a good dirty joke in biology, so I like to have them when I can. Yeah, Actually, I can tell you the joke, I just won't say the punchline until I stop recording. And again, you have to come ask me because it is inappropriate. So if you ask me, we're willing to get something inappropriate. What's the difference between an amino acid and a hormone? Marinate on that. And again, if you want the answer afterwards, I'll give it to you. I should put that in the exam. Anyway, there's a picture of an amino acid. Again, you've got the carbon that's bonded to an amino group and a hydrogen and a carboxyl group, and then of course the side chain, which defines the amino acid, right? Because that's the part that makes it different than the other 19 amino acids. Again, same thing, but it's in writing again. Each amino acid has a unique side chain, giving it its special chemical properties. So here's a good example. You definitely don't need to know these examples, but here's 2 of the 20, right? Leucine and serine. And again, you guys aren't chemists, but you can see that's kind of got like a triangle shape, right? That's kind of got like a square shape. So as you might imagine, that molecule is going to fit together differently than that molecule. Does that make sense? And that's always important when we're talking about molecules, how they fit together with other molecules, but it's especially important when it comes to proteins, as you're going to learn later when we talk about enzymes, right? It's all about how they fit together. So not only do these things have different shapes, and this is a great example, you can see this one's hydrophobic, right? And that one's hydrophilic. So you can imagine this thing, if they were kind of next to each other, that thing would kind of be repelled from that thing, right? They wouldn't want to come together. They repel each other. That, again, is very important. Of all the molecules we talk about, proteins are the most important when it comes to the structure. It's all about the structure. And you can say that for all molecules, but when it comes to proteins, it's even more important. Now, let's make it more complicated. Actually, let me put a big old X to this, just to remind you. Those are just examples. I'm not going to ask you any about leucine or serine. I don't need you to recognize a picture of this and say, oh yeah, that thing must be hydrophobic. And oh yeah, that must be hydrophilic. I'm not going to do that. Though if I was, a little cheat sheet here, generally speaking, it's got that OH on it. You know, we all know that oxygen is very negative. We know that now, but you guys know that now. So that you should associate that with hydrophilic. Anyway, let's make it more complicated. When cells bring these amino acids together, we call those peptide bonds. I don't know why, but somebody decided to give those names, those bonds a name. So it's still a covalent bond, but somebody said, you know what? Screw biology students. Let's make things more complicated. Let's give it yet another name. So if this covalent bond between amino acids, right, it's still a covalent bond, just like all the other ones you ever learned about, what we call them peptide bonds. And honestly, I'm not going to ask you that. And the only reason I even bring it up is because it'll help you hopefully remember this word, which is a word you need to know, polypeptides. So again, the monomer of a protein, those are amino acids, right? And now I'm telling you what the name of the polymer is, and it's called a polypeptide. You need to know that. And that's where it gets its name, right? Polypeptide, because there's a lot of peptide bonds. And here's where it gets even more complicated. I don't know if you guys can tell what this is. It's kind of blurry. Anybody can tell what that picture is? It is, yeah, it is something knitted. Yeah, I guess that's about all you can tell, right? It's a blanket, like a little blanket. It's just knitted. Now, here's where I'm getting at with this. A protein is like something knitted. So actually, let me back up. Yes, you're right. That is something knitted. What else is a way I can describe that? What else is that? Yarn, right? It's just yarn. It's a piece of yarn. But like somebody said back there, it has been knitted. It's still a piece of yarn, right? That blanket is still a piece of yarn, but now we consider it a blanket because it's knitted and it's got that shape of a blanket. Does that make sense? But it's still a yarn. How about this? Hold on. Give it 1 second. Are you kidding me? There we go. Yes. Go. What is this? Piece of paper. Very good. Bear with me. I haven't done this in a while. Okay. What is that? Paper airplane, right? But it's still a piece of paper, right? We can still call this a piece of paper. But now that I folded it in this particular shape, we can also now call it a paper airplane. That is this. So a protein is a polypeptide. And a polypeptide is just a string of amino acids put together. But it's not considered a protein until it's folded into its correct shape. When it's just a string of amino acids, it's just called a polypeptide. But then once it forms its final shape, and shape is very important for proteins, then we call it a protein. It's still a polypeptide, but once it gets that shape, we now consider it a protein. Does that make sense? It's like this piece of paper. This is still a piece of paper, but once I folded it into its particular shape, we now consider it a paper airplane. And of course, it's formed as a function. Same with protein. And again, I realize it's complicated. That's why I'm kind of stalling here. I want to make sure you guys understand the relationship between those terms, polypeptide and protein. So anyway, any questions about that? All right, once again, on the next slide, I'm going to show you two amino acids coming together to form that peptide bond. Does anybody remember the name of that reaction? It's only one of two. If we're bringing something together, if we're bringing two monomers together, two smaller things together, what do we call that reaction? That's okay. You'll need to know eventually. A dehydration reaction, right? So again, whenever we bring things together, we call that a dehydration reaction. Anyway, that's just an example. Are there any questions about that slide? All right, here's a new interesting thing I like to think about. So your body has thousands of different types of proteins, not just thousands of proteins, but thousands of different types. You can look that up for independent work if you want, because that's kind of vague. Exactly how many proteins are there? But anyway, we have thousands of them, but we only have 20 types of amino acids. So how's that possible? And I think the old textbook made this point, not the one you guys are reading. But think about the English language, right? Think about all the different words in the English language. And as of 2016, there was about 1 million of them. All those words from just 26 letters, right? And this is about how we put them together, how we put those letters together in different combinations to make different words. Same thing with proteins, right? We only have those 20 amino acids, but there's so many different ways we can put them together to make all these different proteins. Definitely not going to ask you how many words there are, or how many there were in 2016. But anyway, any questions about this? It's just a good point. I'm not going to ask you any questions about it. All right, if you download the PowerPoint, you can watch this this little animation that talks about the primary structure of protein. Yeah, I'll come back to that. I won't take questions yet. So again, it's the amino acid sequence that determines the three-dimensional structure of the protein, right? So it all depends on which amino acids you're stringing together and in what order you're doing it that gives it its shape, its three-dimensional shape. And again, I'm going to reiterate this here. It's very important. The shape is so important when it comes to proteins. It's all about the shape. Right? Proteins are all about the shape. I mean, there are some other things that we talk about that they contribute to what they do. But of all the biological molecules, when it comes to shape, protein is the most dependent on the shape. Because nearly all proteins work by recognizing and binding to some other kind of molecule. So again, it's all about that shape and how they fit together. Any questions about that?
The shape determines the function.
Mm-hmm. Very much so. And again, that's the case with all molecules, but for proteins, we really focus on that. This is just an example. You don't need to know this, but you can see here that this is a string of amino acids, but because these two sulfurs are bonded together because they're attracted to each other, now instead of having a long straight string of amino acids, it's got a little kink in it, right? It curves back on itself. Just an example, but that just shows you, it depends. If this amino acid wasn't here, and if this amino acid wasn't here, they wouldn't be bonded together, and then this thing wouldn't have that little loop in it, right? It would just be straight. All because we had those two amino acids right there. And then once again, let's make it a little bit more complicated. I don't know if you can read this. Your book talks about this, so you should study it. I'm going to go light on this when it comes to the exam. But when we're talking about proteins, first of all, we have something called the primary protein structure. And you can describe the primary protein structure simply by saying which amino acids are attached to which amino acid, right? So if I told you, all right, we've got this leucine and it's attached to a serine, and that serine is attached to a tryptophan. whatever, right? If I'm just describing which one is attached to which, I'm describing the primary structure. Then we have something called the secondary protein structure, and this might be hard for you to read. Hydrogen bonding of the peptide backbone causes the amino acids to fold into repeating patterns. So instead of me just telling you which amino acids are attached to which, if I'm describing how they're folded, right, then I'm talking about secondary protein structure. Then we have tertiary protein structure, and that's the three-dimensional folding pattern of a protein due to its side chain interactions. So as far as we're concerned, secondary and tertiary are pretty much the same. One's about hydrogen bonds, the other one's about side chains. This is a 100 level biology class, 100 level class for non-biology majors. So as far as I'm concerned, this is not important to you. If you brain dump everything that's on the slide, you will understand things I'm going to teach you later, right? So that's why, in my opinion, it's not important. And then finally, just because it's up here and because it's in your textbook, at the very bottom there, we have quaternary protein structure, and that is protein consisting of more than one amino acid side chain. In other words, if we're talking about the quaternary structure, sometimes proteins are made up of different polypeptides. So we might have this one, and then this one, you put them together, and now we have a protein. But again, as far as we're concerned, this is useless. It's in your textbook though, so I wanted to bring it up. So when you're reading your textbook and you're studying and you're getting overwhelmed with that part, just remember, I'm not gonna ask you any questions about that. You can not understand this, but still understand the things that you're gonna learn later. And to me, that's what's most important. Anyway, some more examples. So you can see here, and maybe you can't, it's blurry. You can see each one of those amino acids. This is telling me which one is which. And again, that's describing the primary structure. Because all it's doing is telling me which is which. And then of course, when they get folded the right way, now we have an actual protein instead of just a polypeptide. And this is a picture of the same protein as that. And the only thing I'm getting at there, once again, in biology, we have different ways of showing things, right? Any questions about that slide, which is just an example? All right. So as you might imagine, again, the form is very important for these proteins. So if you have a slight change in the amino acid sequence, even a slight one, that's going to affect the protein's ability to function. If you change just one amino acid, especially if it's an important one, you're going to change the shape. And if you change the shape, you change its function. This will be very important later in the semester. We're going to talk about mutations. And without mutations, we wouldn't have evolution. And without evolution, everything would just be a cell, right? We wouldn't exist. Anyway, a good example of this, and I'm not going to test you on this, but it is a good example. Hemoglobin. So have you guys heard of sickle cell disease? Anyway, sickle cell disease is caused by just one amino acid being different. So that's what a normal red blood cell looks like. This is what a sickle cell red blood cell looks like. Sometimes it's deadly for people. It's a serious disease, all because of that one amino acid was changed, right? You change one amino acid, and now all of a sudden you have a whole different shape of a protein, which causes a whole disease. And we'll actually come back to this, because again, later in the semester, we're going to talk about mutations, and this is a great example of mutation. Because what I haven't taught you yet is what determines which amino acid is attached to which amino acid, and we'll learn that later. And if you change that thing that determines it, then you change this, and now all of a sudden you have disease. So again, everything I just said, here it is in writing. One difference in the sequence of amino acids is enough to cause a protein to fold into a different shape than it should be, right? Which alters its function and sometimes can cause diseases. So think about things like Alzheimer's, for example, and that's not even on here, but that is a disease of proteins. The proteins in your brain are misshapen. Then there's also something called prions, which I'm going to talk about very quickly. You've probably heard about one of them. Mad cow disease. Have you guys ever heard of that? That's what we call it, BSC, but we call it mad cow disease. How many of you have heard of Kuru? Might look that up for independent work if you want. That's interesting. Guess how that spread? I mean, you can tell it's for humans, right? But guess how that spread? Cannibalism. People eating other people's brains, they get Kuru. And there's a place in the Pacific where that's more prevalent than other places because they eat other people's brains. There's a bunch of different examples, but then you can see fatal weight loss in deer, elk, and moose. There's a bunch of them. You can look them up for independent work. So again, those are just 3 examples, but there's others that you can look up for independent. And this is what I like how it kind of shows what it looks like, right? So this is a normal brain. We have these things called prions, which are just basically misfolded proteins. But when they come in contact with other proteins, and they cause those proteins to become misfolded. So then you literally get like holes in your brain, which is where you get spongiform, right? That's where that word comes from. It's like your brain turns into a sponge. That's a good place to stop. So when we get back, we'll talk about what causes these things to be misshapen. And I guess the last word for attendance today will be Monday. So you guys have a good week. Like I said, I've got a lot of catching up to do with grading, so I apologize in advance to be slow to grade your stuff and return your emails. Yeah. Okay. Good, yeah. Let's do it on Friday. Yeah. For lab 4? Yeah. Yes. So lab 3, the lab itself is just the questions, but yes, there are links. So if you click the link, then that brings you to the instructions on how to do the experiment. Yes. Yeah, exactly. So when we do the experiment and take the data, this is where we're going to go. Water there. Don't forget to do this. Just a quick reminder. Mhm. If I remember correctly, it's not extra credit, it's part of the pre-lab.
Okay,
If it is extra credit, it'll tell you, it'll say, hey, this is extra credit, but as far as I remember, it's required. And also, if it's required, it'll tell you that too.
Okay, and one more thing is, you've been mentioning independent work.
Uh-huh.
Is that going to be questions from like a PowerPoint?
No, that's anything you want to write about that has to do with biology.
Okay,
And when I'm saying, when I bring it up in class, what I mean is like, that's just suggestions. Like, oh, since we're talking about this, you can look it up for independent work if you want. But it could be anything. Like if you're into cats, for example, you could write a paper about cats.
because I was thinking, is this like part of extra credit?
No,
Or is
no.
it like optional or something?
Not at all. So if you look at the syllabus,
Uh-huh.
it's worth 100 points. And the whole course is worth 1,000, so it's a whole nother grade.
Okay,
So definitely not optional, definitely important. And
okay, just because, I did one of these, like semester,
Yep.
and I want to do it from the other, like, PowerPoint stuff. If I could.
You can.
So I was just thinking, hopefully, I could actually,
Yeah. And if not, like on the last one, if you make mistakes, I'll just leave comments and say, this is where it can be improved, or here's something you have to fix before I can grade it. So no pressure. At least if you get started now. And if you wait until the due date to turn it all in, that's high pressure because if you screw it up then, you're out of time to fix it. So
But yeah, I was just wondering, these charts are here and this one up here, that's for the actual funding.
Yep.
Website. Okay, so
correct.
there's no link to it at all. Okay.
correct. Yes, sir.
So for my lab too, you told me to do the online version.
Yes.
So only have to do like questions 22 to 28, I think.
I don't remember the numbers. You definitely have to do the whole thing. The only difference between the online version of the lab and the one everyone else did is we physically put acid into some things. We watched how the pH changed. But for the online lab, it's completely different for the pH part. And on that one, it's just like, hey, what do you think the pH of battery acid is? And you guess, and then you look it up. Yeah, and then you look it up like, was I right or was I wrong?
And I already got like the whole lab done. I just have to do that last little part.
Okay.
So can I just determine like the online version, like part of the PDF thing?
Yeah.
I don't know how to explain it. I want to send 2 in. Like I already sent one in, and I'm also going to send the questions I missed from the online lab.
Okay. Perfect.
And it is.
If you need help, I can be online to help you.
Yes, sir. And then the pre-lab for lab 3 is already due.
Yes.
It was due Friday.
Yes.